Good evening and welcome. My name is Jim McManus, and I am the new Vice President for Administration and Operations here at the lab. And now I want to introduce our speakers. Maya Groner joined the lab in about November of 2021. And Maya came to us she got her PhD at the University of Pittsburgh. She then went out to Prince Edward Island at the Atlantic Veterinary College of the University of Prince Edward Island, then went to the Virginia Institute of Marine Sciences, and then went to Prince William Sound Science Center before joining Bigelow. The two folks that will be talking with Maya today, Reyn Yoshioka joined Bigelow in June of 2022. Reyn might not know this, but I went to Oregon State University, so the fact that he got his PhD from the University of Oregon, I take note right away. Not that there's any animosity that Reyn knows about. And finally, last but not least, Melissa Rocker. She got her PhD. Melissa, so both Maya and Reyn were like far out west before coming here. Melissa was like in Australia. So Melissa got her PhD from James Cook University and the Australian Institute of Marine Science. Prior to Bigelow, after her PhD, she stayed in Australia, was at Deakin University and the Australian Institute of Marine Science.
Well, thank you all for coming out. It's such a beautiful day outside, so extra appreciation for your willingness to be inside to learn about marine diseases in our oceans today. So I run the Quantitative Marine Disease Ecology Lab here. We call ourselves Q Model, rolls off the tongue. And the lab is really interested in understanding what drives diseases in our oceans and what the consequences of those diseases are. And it's not just me, as Jim mentioned, we have, let's see if I can get this pointer to work. We've got Melissa here, who came from Australia. She's a postdoc in the lab. Reyn Yoshioka, who was a postdoc in the lab and has recently been promoted to a research scientist at Bigelow. And then Kirsten Johnston, who is a research technician. And so this collective group has done this research that you're going to hear about, along with a handful of, the dogs provided a lot of moral support. Yes, they helped, always. Yes, so the interns also helped with a lot of this work. So we're interested in diseases in our marine environments, and just like you all experience disease yourselves in terrestrial environments, the oceans have a lot of infectious diseases, and some of these are high impact that we should be concerned about, and some might just be merely kind of annoying or maybe super interesting if you're excited about unusual biology. So in this case, this is a picture of a shrimp that is, that's not an egg sac of the shrimp. It's actually an egg sac of a parasitic barnacle called a rhizocephalin. So the barnacle has actually, it's taking nutrients out of the shrimp, and then it creates this big egg sac that the shrimp takes care of as if it were its own eggs. And then the eggs will, the sac will open and it will release new larval parasitic barnacles, and the cycle will repeat itself. So this barnacle can actually feminize the shrimp so that even male shrimp will take care of these eggs as if they were their own. So it's super interesting, not terribly problematic. We see this in very high abundances in very healthy shrimp populations. In contrast, you might worry more about this disease, which is, this is a seal that has died from bird flu, or highly pathogenic avian influenza. And we think of this as a disease that infects many species of birds, but it also infects mammals. You've probably heard about cattle with bird flu, but marine mammals, especially seals and sea lions, are very susceptible to this disease. And we're seeing mammal-to-mammal transmission, which is a concern for us as a potential spillover to humans. So this has been documented in Maine. About 60 seal deaths, I think, have been attributed to bird flu. But it's really problematic in the southern hemisphere, and there are locations where there have been tens of thousands of mortalities of seals and sea lions. So something to keep an eye on, and maybe a reason to appreciate the impacts, or at least respect the impacts of marine diseases. Another one you might have heard about, and it was in the news a lot this week, is sea star wasting disease. So this is a disease that was really emerged in 2013, so a little over a decade ago, and caused the mortality of billions of sea stars, of more than 20 species. This sea star here is, it's called a sunflower star. It's the biggest sea star we have in the world, and it was so impacted that is now critically endangered by the IUCN. So very high impact disease. I'm supposed to stand between these red lines, so I think I'll scoot over when I get the signal. So again, just demonstrating that these diseases can have really high impacts, and we really need to understand what's causing them, and then potentially get to the point where we can think about management in some cases. So in my lab, one of the first questions we think about is, what is causing disease? If we see a mortality event like this, we want to know, is it environmental? Is it a pathogen? This is a number of Pacific razor clams that were found littered across a beach in Washington State. And we developed a diagnostic test. So this is a slide, a thin section of the gill tissue of these clams. And there's actually a DNA-specific probe that is causing these dark dots on here that's binding to a bacterial pathogen that infects the gill tissue. So we have a diagnostic tool that we can take and sample clams like this and determine what the cause of mortality is. So that's one kind of question we ask. We also think about, well, is something changing that might be increasing the amount of disease we're seeing in our environment? And climate change is kind of the obvious culprit, right? We're seeing, especially in the Gulf of Maine, really rapid warming of not just sea surface, but also sea bottom temperatures. And it's sometimes heat waves. You've probably heard about the heat waves that we're getting in the oceans and on land. And these can change the physiology of both the host and the pathogen and sort of tip the balance in different ways. So we might see the same diseases that have been in places in the past, but just having sort of different patterns, different levels of mortality, different timing, and that could be triggered by changes in temperature. So one example of this is with a species that I know folks like to fish here, which is the striped bass. This is a picture of one from the Chesapeake Bay that has severe mycobacteriosis. And if you're going gross, that's actually an evolved response that you have that's called a disgust response that helps you stay away from disease. So side note, so this mycobacteriosis, it's a relative of tuberculosis, and it causes these very slow progressing infections in striped bass. And it's very temperature sensitive. So in the Chesapeake Bay, we were able to model this mortality due to disease of this fish. And we found that at about 26 Celsius, which is about 80 degrees Fahrenheit, we saw about 40% survival, which isn't great for a very diseased fish, but it honestly doesn't look like it would even survive that long. But then when the temperature gets to about 85 Fahrenheit, which is about 29 Celsius, the survival goes down to about 10%. So, and these are temperatures we're seeing in their main spawning ground in the Chesapeake Bay. So they're right at this tipping point. And so thinking about, even it's not a huge change in temperature, just 1.5 degrees Celsius, but it really has huge implications for a disease like this. So we also want to think about the impacts of disease. And when we do this, I think a lot about the host. So who's the host and what role do they play in the ecology of a system? So if it's something like this seagrass meadow that provides habitat for lots of young fishes and it sequesters carbon and protects coastlines from storms, we're going to be very worried if we see diseases that are causing mortality in a seagrass meadow. Similarly, if it's a lucrative fishery that supports an economy like the snow crab, we also could be concerned about potential impacts of disease. So speaking of fisheries, in the US, some of our highest valued species are crustaceans, so crabs and lobster. And that's what we're going to hear about today from Melissa and Reyn. So 2 projects that we have. One is all the way in the Bering Sea in Alaska, looking at a disease of snow crab. And the other one is here in the Gulf of Maine, looking at a disease of lobster. And you'll see how despite being very far away, there's some very common connections and parallels in the way what's driving these diseases, the way we study them, and the way we go about our research process, which I hope will be very educational and exciting for you all. So first, we're going to look at this question: how is disease impacting some of our most valuable and iconic fisheries? And Reyn is going to be taking us on a journey backwards to see what we can learn about by looking historically at what's happened, not too far back in time, just about to 2017, and what we can learn by studying patterns of disease in the past. And then Melissa is going to help us look forward and think about how can we predict disease into the future and increase preparedness, and how do we get to that point? So I will turn it over to Reyn.
Thanks, Maya. So my section is going to start off with a journey that just began only a week after I moved from coastal Oregon, go Ducks, to coastal Maine. So a week after, I flew back to the other coast to get to Alaska, where I was curious about where I was going to find some food. It turns out this place where I was at, Dutch Harbor, which is a major seafood processing port, has actually quite a few food options that are pretty great. But unfortunately, the food isn't what I'm going to be talking about today. I was really there to get onto the Eastern Bering Sea Bottom Trawl Survey, which looks at this area over here, which is the Eastern Bering Sea. This is a survey that's conducted by NOAA every single year. Now, the Bering Sea, you can think of it as a really cold environment. So you see all of the ice on this image that really typifies the environment and controls the species that lives there and the kinds of ecology that they have. One of those really cold-loving species is the snow crab, Cainicides opilio. They're delicious, but also they have great harvests and are really lucrative. And so you can imagine the importance of those fisheries to the local communities that rely on them. In thinking about them as being another crustacean, you can liken them to Maine's lobster in terms of their importance for Bering Sea Aleutian Island communities. Now, I wasn't here just to look at the snow crab, unfortunately. I was there to look at this emerging syndrome called black eye syndrome. If I accidentally slip into BES, that's what I'm referring to. I will try not to, just a warning. But we're super creative with the names. So when in 2017, folks at the Alaska Department of Fish and Game first observed crab that they had in the lab, and then also crab that they were bringing back from surveys with these black marks in their eyes. And so it was hence dubbed black eye syndrome. A typical healthy crab eye looks like this. We're looking at it right in the middle. Don't zoom in with your eyes too much if you don't like to look at bug eyes and their compound eyes, because they're about the same thing. But I think they're really pretty. So they have like this kind of like sparkly, multifaceted appearance. There isn't really any dark marks on there, which do appear at these early stages of black eye syndrome. So you start to see these dark marks appear inside of the eye. Sometimes they can grow and coalesce until the entire eye is black. But at these stages, there isn't really clear damage that's happening to the eye. Now, that's very different when you get to the severe stages, where the actual surface of the eye gets impacted, and then it can start to erode away and then eat back into the eye stalk. So this crab over here is not doing well. It can't see very well at all, because that visual surface of the eye is completely eaten away. Now, if we want to think about kind of what that looks like on a microscopic level, Maya mentioned before the thin sections of tissues that we can get. This is called histology. And so basically, it's a way for us to look at the tissues of an organism. And it's really important when we're doing disease work to see what kind of damage is happening at that really fine scale. So essentially what happens with this is that we get the crab, we preserve their eye stalks, we ship them over to a colleague, in particular one at the Virginia Institute of Marine Sciences, who then takes the eye stalks and cuts really thin sections of them. Think like prosciutto, but like even thinner. And so he then mounts it onto a slide and then applies different kinds of dyes to it. And that's where these purple and pink colors are coming from. So that's to highlight different kinds of structures that we see. So a healthy eye over here has mainly the cornea, which is the eye surface. You can see these nice two layers that come out of it. Very nicely intact, pretty uniform in size. And then right beneath that are the ommatidia, and these are the seeing structures or the seeing cells. And these are those individual components that in a compound eye come together to create the image for that organism. So that looks great. It looks to me organized. It might just look like a bunch of pink and purple to other folks, which is just as well, but that really contrasts with what a severe black eye looks like. So that's this over here. The cornea up on top is super damaged and ragged. You can see it's kind of frayed. And then the ommatidia are kind of retracted back. And then they're also, in some cases, just gone. So this crab very much cannot see. So not a good time if you're a crab having severe BES. Something that we do when we take histological sections is try to figure out if there might be a microbe or something that might be causing that particular syndrome, especially if it's one here where we don't know where it's come from or what causes it. Unfortunately, there's no histological evidence of a pathogen, so we actually don't know what causes black eye syndrome. What we do know, though, is that in the years preceding the first observations of black eye syndrome, so 2017, there was a big heat wave in the North Pacific from 2014 through 2016. This was known as the blob. What this is showing is a map of temperature anomalies. So that's when water is unusually warm or unusually cold compared to the long-term average. If it's warmer colors, like these reds, it means that the water is unusually warm. And if you look up at the Bering Sea, it looks like it's on fire. And so even if this is a warm month in September, you can see just how devastatingly warm it was. And for somewhere that's supposed to be dominated by sea ice, you can imagine the big shifts in the local ecosystem. And we think that that could have effects on the snow crab. So if you're looking at emerging syndrome, it takes a lot of different kinds of expertise to understand what's going on. That's because you need folks who are really tuned into the pathology. So that's like how disease presents on an individual level and the damage that it does to tissues, scaling up to folks that work on the ecology of diseases and then the larger communities around them. And then also taking things, when you think about fisheries, all of the human communities that also rely on them. So the team that we have that's supported by the North Pacific Research Board really tries to capture that broad range of expertise that is needed to understand marine diseases. Similarly, we have to look at marine diseases from a bunch of different angles. I was up in Alaska to do some in-field sampling and then also to collect crab for a lab experiment that we had in Kodiak. But really what I'm going to be talking about is statistical modeling. And so that's when we take a data set and then try to understand the relationships in it using fun computers and math that I'm not going to talk about too much. But the short of it is when you have good data, you can make some really cool models. And so these data are from the same bottom trawl survey that I participated on, but going back decades. And so this data set has been produced by NOAA through all these years over their entire survey grid. So it's a huge amount of data covering large space and time. And this is showing the snow crab abundance through time, also in relation to temperature. So don't dwell too much on it, but pretty much what you should be taking out of it is all of the blues, all of those colds, are times and places where there's a lot of snow crab, and then the reds are when there are less snow crab. And so this really highlights the fact that when we have these good data sets, we can really interrogate those relationships, but also that snow crab, unsurprising to their name, like cold water. Now, disease data is a little bit trickier. So there's often not a lot of collection, and that gets even more complicated if you're working with a deep or remote species. Similarly, with environmental data, if you're working with something that lives on the bottom, those data can be hard to come by. So at the surface, we can go directly measure the temperature of the water, or we can use things like remote sensing, so using a satellite to understand the surface of the water. But if things are deeper in the water or on the bottom, you can't really do that. So what do? So this is where the Alaska Department of Fish and Game and the University of Washington Cooperative Institute for Climate, Ocean, and Ecosystem Studies come in. So through them, we were able to get some really great data sets. The first is the Alaska Department of Fish and Game's Crab Observer Program. So they're essentially fisheries observers that go out on the crabbing vessels and assess portions of the catch so that we can better understand the species through time. This was super exciting for us because through this effort, they were able to collect data on 700,000 crab. So for me, I'm happy if my data set has like a few hundred individuals. So getting this data set was pretty much a gold mine. This happened over the span of 4 years, starting in 2018. So that's only just a year after black eye syndrome was first observed. So that's a really quick turnaround in data collection. That's often really hard to do. And the only reason that it's only 4 years is because the latter years were actually closed to the fishery for reasons that I'm going to talk about in a bit. Really excitingly, though, this is integrated into and includes data of established monitoring. So that means we have all of this biological data that goes along with the crab that we can relate to black eye syndrome. So for the environmental data, we are harnessing an oceanographic model. In particular, it's the Bering 10K ROMS, if you want to be nerdy about it. The short of it is that this oceanographic model tries to imitate the physics and dynamics of the ocean. And by benchmarking that against the real data, you can get estimates of what the bottom conditions look like for things like temperature, pH, all sorts of different variables on a much finer spatial and time scale than you would be able to get with direct measurements. So this was also really exciting to work with. And so when you stitch these two data sets together, you get me happily playing around and poking at the data, trying to code something up. Sometimes coding doesn't go as well and you spend a lot of time debugging, but eventually you get to a model that you can work with and you can start to try to understand what's going on. So up over here, these are 100 crab. So you can think of this as representing one crab being 1% of the population. And so if we look at kind of what black eye syndrome looked like, in a low year, it was only about 1%. So not much of the population is affected. Even in a high year, it was only about 4%. So that's pretty promising, right? It's not a super, super common syndrome that's going on. What I do want to highlight, though, is that diseases and emerging syndromes tend to be really patchy. And so you can get these hotspots where, in this case, it was 38% of the population in a particular location in time had black eye syndrome. And so if you don't have these really robust data sets that are going out over multiple years and multiple time points, you can often lose these kinds of dynamics. But if we look at kind of the relationships that we're able to find, the first one is that crab that have older shells were much more likely to have black eye. Similarly, crab that are larger also were more likely to have black eye. This is going from the minimum legal size to the market preferred size. And then also, if you increase the density of crab, you see more black eye syndrome. Now, what we're really interested in was this temperature effect. So if you go from 2 degrees Celsius to 3 degrees Celsius, so it's only about 2 Fahrenheit, you see a pretty substantial increase in the probability of black eye syndrome. And that's pretty startling, right? This is still in the 30s. It's still pretty cold water. It's not that much of a difference in temperature, but we see that increase by 68%. So that's all interesting, and it's good that we're able to understand black eye syndrome a little bit more, even if we still don't know what causes it. But this is also set in the context of the collapse of the snow crab stock in the Bering Sea. So between 2019 and 2021, there were an estimated 10 billion crab lost in the stock. So that's absolutely catastrophic, right? All of these individuals have basically disappeared from the communities and the other organisms that they interact with. And then you can imagine how devastating that is for the communities that rely on them. So this actually ended up triggering the closure of the snow crab harvest in Alaska for the first time in history. For us, and I hate to call this a silver lining, but I think it's interesting, is that this actually meant that our crab that we were studying made it onto national news, but this is never the reason that you want your crab to make it onto national news. So the hands that are holding this is actually our colleague at the Alaska Department of Fish and Game, holding one of our crab from the temperature experiment that we ran for black eye syndrome when he was interviewed by CBS. So if we look at the collapse of the population, as well as what we know about BS, we see some interesting parallels. So folks looking into the collapse, particularly at NOAA, who actually published the paper on this, have come to the conclusion that it's most likely a mass starvation event. So the way that that happened is that they saw much higher densities or abundances of crab in the years preceding the collapse. And this also coincided with a marine heat wave. So not the same one in 2014 through 2016, but it happened again in 2018, 2019, and it was also record breaking. But because these crab are cold-blooded, their metabolisms rely on the outside water temperature, and so those got ramped up. At the same time, because these crab really like these cold environments, they got even more crowded together because there was less of that cold water for them to hang out in. And so all of that together means that there are a lot of hungry crab, energy demand outpaces energy supply, and when that gets really bad, you can have a mass starvation event that could lead to the collapse of the fishery. So if we think about black eye syndrome, we saw that crowded crab and then also warm water ended up with more black eye. When we looked at the crab that we had in our experiment, we actually saw that ones that had black eye had lower energy reserves than those that were healthy. And so this also points to a potential condition effect. And so we think that black eye could be an indicator of crab in distress. For one, we don't think that black eye itself is responsible for the collapse. We didn't see that much black eye syndrome rate, only 1 to 4%-ish. And then also those most severe cases where we think that it's going to be really bad for the crab, were even just a fraction of those. But the collapse, as well as what we see in our black eye data, suggests that they might have similar causes, and it really highlights the fact that these crab were probably not doing well in this period of time. And so there's a couple takeaways that I hope that you get from what I'm talking about. The first one is the importance of agencies and these long-term data sets. So this monitoring effort by the Alaska Department of Fish and Game, the Crab Observer Program, as well as the NOAA bottom trawl surveys, create these long-term rich data sets that we can use and to try to understand the past. Similarly, modeling efforts are really vital because if we can't get at all of those data in the past, how are we supposed to understand what's going on? Especially so, these are hard to do. It's really hard to model the ocean, if you can imagine. And then also going out in the Bering Sea during the winter fishing season, if you're a crab observer, to assess portions of the catch is not an easy feat, right? I think I would not do too well in it. So huge props goes out to these folks for collecting these data sets, because these are not the flashy things that folks tend to think about. But the only reason that I'm able to talk to you about our results today is because of these efforts that have gone in. And then the last bit is that the relationship that we saw with black eye syndrome and the warming seas really adds to that growing body of evidence that highlights the fact that we need to be keeping an eye on our marine species and their health in these changing climates and in these warming waters, right? We still don't know what causes black eye syndrome, but we see a relationship to the temperature, which really means that we need to be vigilant and encourage more monitoring of marine health as we move forward. So with that, it's time for questions.
Questions on here?
Thank you very much. Very interesting talk. The fact that you have a crowding effect suggests that a pathogen may be transmitted. And if so, is the thought that it's a bacterial or a viral agent? And maybe the resistance of the crab to such an onslaught may be weakened with temperature. But it's dramatic. The temperature effect is really dramatic.
Yeah, that's a great question and point. That's been one of the perplexing things about black eye syndrome is at least the histology hasn't pointed out a potential pathogen. We do have a colleague that's looking at using DNA to try and see if there might be differences in the microbes that are present, but we don't have that information yet. And so that density effect could be also related to transmission, which was really perplexing when we originally thought about it and we are like, we don't have a candidate pathogen, so how might this be playing out? So yeah, density can work in a lot of different ways, and this is the one that we have our kind of best candidate with.
One more question, right, Leah? All right.
When the crab has black eye syndrome, can you still eat it? And what happens to these crabs? Do they fall to the bottom of the ocean dead, or does something else eat them?
So we know that snow crab do rely on vision. That kind of surprised me because I thought that they would be deep and they wouldn't need it that much. But they do see quite well, which is surprising to me. Maybe not surprising to other folks, but anywho's, as far as I know, I would be perfectly comfortable eating a crab with black eye syndrome. We actually don't have any direct evidence for mortality that's associated with the syndrome. But I could imagine that if a crab can't see that well, it's going to have a hard time sensing predators, and it's also going to have a hard time sensing food. So it might be a different story kind of in the field, what black eye syndrome means for a crab.
Okay, we'll turn the microphone over to Melissa.
Thank you guys so much for coming, and thank you, Reyn and Maya, for such a great introduction to disease ecology and the snow crabs. My name is Melissa Rocker, and as Jim said, I'm a postdoctoral scientist at Bigelow. I'm really excited about this opportunity to talk to y'all today regarding some of the research that I'm working on within Q-Model. And as you can see from these photos, you always get my best side whenever I'm working. But I love looking into tanks and learning new things about the health of our oceans and the marine organisms that we really depend on. So from measures of coral reef and coral health in Townsville on the Great Barrier Reef, down to Southern Australia, where I worked at Deakin, looking at fish health and fish nutrition from more of an aquaculture perspective, all the way to way over here in Maine, where I'm getting to look at disease within our own backyard in the Gulf of Maine lobsters. So let's talk about the health of these lobsters in the Gulf of Maine. And I want to start with a question. Does this look like a catch you would like to eat? I mean, it is getting close to dinner time, so yeah, are we digging this? I would be pretty keen to get in there, but I do want to zoom in just a little bit and show you this one guy. So this is a lobster that actually has what's called epizootic shell disease. And it's a pretty severe case at that. And so you might be wondering, what is epizootic shell disease? Well, first of all, I want to let you know it is not contagious to humans, and it's actually not contagious from lobster to lobster. It isn't like COVID during the pandemic, where it was about proximity and spreading between individuals. So epizootic shell disease is actually a real concern for the lobster industry, in particular because it makes the lobsters ugly. And in the live lobster trade, which is what most of the lobster industry is based on, you can't get money for an ugly lobster. And I actually have a few, could I get y'all, shells with epizootic shell disease that I'll pass around so y'all can get a visual of what it actually looks like on the lobster shell. So ESD, actually appeared in southern New England waters, so a little bit south of us, in the late 1990s. And it's a dysbiosis or an imbalance of the bacterial community on the shell of the lobster. And it actually sort of digests down the shell. You can think about it as similar to your gut microbiome. It's a microbiome, but it's external to the animal. And so when that microbiome goes out of whack, you start to get this degradation. And it visualizes as pitting and erosion, and it can happen on the shell, the tails, the claws. And what we can see here is actually a visual of this from some of our own summer interns last year that they put together for us. Annika and Ashley, they did a great job working on this project with us. And what you can see on the left is a lobster with no disease, and it progresses through the 4 stages that our fishing industry as well as managers use to qualify what stage of disease the lobster's at. And so this condition is actually sort of thought of as an opportunistic disease, and it's definitely facilitated and driven by environmental stressors, and in particular, temperature. So what does temperature do? Well, first off, temperature drives increases in ESD. At about 12 degrees Celsius or 54 Fahrenheit, we can start to see some of those first initial signs, maybe a little pit here or there. But as the temperature progresses up and higher, when you get into balmy 20-degree water, which for us that might not be that warm, or 68 Fahrenheit, you really get widespread severe disease occurring. The other thing that temperature can do in terms of lobster biology is it drives their seasonal patterns in molting. So in order for lobsters to grow, they grow incrementally and only once about a year in adult lobsters where they actually shed their shell. And what you can see on the bottom is a visual of this occurring in a lobster that we had during our experiment. On the far left, you see the lobster. We know he's got a bit of an older shell because he has severe disease, but they actually sort of split at the base of the carapace and the top of the tail and butt their way out of that old shell into a new soft shell to begin with. And over the next days to weeks, it starts to harden up so that they've grown in size. And it turns out that with epizootic shell disease, the lobsters actually can die from the disease itself if it gets severe enough, but they can also die during this molting event if there are complications in trying to remove themselves from that old shell. So while ESD can affect mortality, we also know that these lobsters can actually survive through ESD because some of these new shells actually have little scars that indicate survival through out of a diseased shell. So in essence, temperature really is important in looking at the dynamics of this disease and in what the lobsters are doing each year. So within the New England area, we have 2 populations, 2 stocks of American lobster. We have the southern New England population, which is down near New York, Connecticut, Rhode Island, Mass. And then we have the Gulf of Maine population, which is further up by us. And these are managed separately. So what we have here is a graph of lobster population, their abundance across the years. And you can see I've starred right above the late 1990s. That's when we started seeing ESD first appear. And you can see by the early 2000s, there is a crash in this population. This crash was concurrent with rises in temperature. So really getting those ocean waters within sort of the southern New England population heated up. And it really brought the whole population to its knees. What's interesting is that in concurrent with these rising temperatures, we also had an increase in epizootic shell disease. It was as high as 60, sort of averaged across the whole population, but you could once again see hotspots with up to 100% coverage within the population. Now, on the other hand, we have the Gulf of Maine, which here we once again have our lobster abundance across time. And what you can see here is in the late 2010s, we had this exponential increase in the lobster population in the Gulf of Maine. That was about the same time we started getting some warmer waters progressing up into the Gulf of Maine. And so we're thinking maybe that's a really great temperature for the lobsters. It's a recruitment optimum or a physiological optimum where they were at a happy reproductive point. But that begs the question, as the temperature continues to rise, what will happen to the lobsters in the Gulf of Maine? Are they fated to have the same outcomes as their southern New England counterparts? And how will epizootic shell disease sort of present? Will the effects be similar to southern New England, or will something happen where there are different effects based on these different populations? So with these questions in mind, we brought together the A team and collaborated with some state agencies and scientists at the state agencies and received federal funding from Sea Grant. In particular, we worked with Tracy Pugh at Division of Marine Fisheries in Massachusetts. She brought all her amazing knowledge about the southern New England population and the lobster biology. And we also worked with Kathleen Reardon and Heather Glahn just down the road at DMR, Department of Marine Resources, on the other side of Booth Bay. And we had our amazing team from Bigelow to really bring that collaboration together, where we had people who could work on tank experiments, people who could model and use oceanographic data to really tie this whole project together. And I think, I forgot to mention, so working with Kathleen and Heather was really great because they are really imbibed into the lobster industry, and they have really close connections. And by working with them, we were able to get at the questions in terms of disease that lobstermen were keen to look into and have answered. So we set up this tank experiment, and the question was, how does temperature affect the disease progression? And within that, how does the disease progress if you use different lobster populations? So we set up this common garden experiment at Maine DMR using their wonderful wet lab facilities. And we used lobsters from 3 different parts of the Gulf of Maine and southern New England. We had Maine lobsters, we had Massachusetts lobsters, and Connecticut lobsters. And we took 62 male lobsters and split them into 2 temperatures. We used that 20 degrees Celsius where we had that widespread high disease severity, and we used 12 degrees where we might start to see a few little signs of disease. We put these lobsters randomly into individual tanks to segregate them, and we used lobsters that were just shy of legal size at 80 millimeters. This way, when they molted, if they molted, which they did, they would be entering into the fishery. They would be relevant to the fishery and what you actually would have caught that year. We ran this experiment for 140 days. It was a long experiment, monitoring them daily, but taking measurements and taking photographs every 30-ish days to really see how things progress based on temperature and population. And Maya and Reyn showed some beautiful histology, and at the very least, if you're not into histology, that's okay, but I would hang these in my house. I just think they're so pretty. So, but what I have actually on the left is a picture of a histological image of a disease of a lobster with no disease. And what I really want you to take away from this is that the bottom half is the soft tissues inside, but the top half is actually the shell. And it's the EX, or the purple line, is the outer cuticle, and the pink is the endocuticle, or the inner layer. And these layers are beautiful and crisp and clean, similar and all the way across the image. Now, when we sampled our lobster, we lethally sampled them so we could get some of these images. And this is actually an image from one of our diseased lobsters. And what I really want to point out is that, so we're zoomed in more on where the outer cuticle and the inner cuticle are, but the outer cuticle has pretty much disappeared. And those new purple blobs are actually aggregations of bacteria. It's the bacteria kind of worming its way down into this lattice, these pillars that have been degraded of that inner cuticle. So you're really seeing the destruction of both that outer and inner shell working its way down into what will eventually come into the soft tissues inside. So when we ran this experiment, we had 3 main findings that I'm going to focus on that'll lead us into the next section. But when we exposed our Maine lobsters to warm water, we found that these lobsters developed more severe disease. So they had higher coverage of disease on their shells. We also found that the disease progressed faster. So they went from less disease coverage to higher disease coverage quicker than their southern New England counterparts. But finally, these lobsters also were found more likely to molt. So they were more likely to use their energy reserves and push that easy button and get rid of that shell disease. So with that, we then want to create a model that can really look to the future and tell us what we might see in our Gulf of Maine as we progress forward. So by doing that, I've been working with the team to develop an individual-based model. And this is a state chart from that model. So we essentially pretend we have one lobster. This lobster enters the model into a disease-free state. There's some rate of infection that occurs that we got from our experiment that he can progress through mild, moderate, and severe disease at different progression rates depending. And then at any point within this model, this lobster is able to molt out and recover back to a healthy shell. But there's also a chance that he could potentially die and have a mortality event and not survive that molting. So what we did is we put this one lobster and created a population of 500. So I've created this model simulation video that I want to show y'all. So here on the left, I have my hypothetical lobster population, and there are 500 individuals, and they are all green at a healthy state. The top graph actually shows you the lobster population, their percent of disease within that population, and they can go from mild to severe to moderate or even dead, or back to green. And then across the bottom, we have a temperature profile from our own Booth Bay waters that shows 0 to 20 degrees in temperature, Celsius. And all this is occurring across days of the year. And what we can see is right about day 200, which would be July, mid-July, when the temperature is increasing, we start to get that summer disease event. And we see we have mild and moderate cases begin. And as the year progresses, we get into the second year, and around day 500, 525, all the disease disappears. This is actually the molting event occurring that we've put into the model, that annual event that all the lobsters go through in order to grow. And as you see across time, we can cycle this profile and look at changes in temperatures and see the population and see the disease come back, and the lobsters molt out of disease over and over. And I think this is a really, really cool visual tool. It helps us communicate our work to y'all, to the public, but I also think it'll be really great to communicate with managers who have questions and fishers who may say, well, what happens when the water gets colder? Warmer? What happens if the water's colder? What happens if we have more molting events each year? We can change these parameters within this model and sort of see different outcomes and really predict that ESD risk out into the future. And so with that, I will pass back to Maya.
Well, that applause was for you, Melissa. I can't tell you how lucky I am to have these two in my lab working on these amazing models and helping us learn so much about what climate change is doing to disease in these fisheries. So I'm just going to summarize some of the lessons we've learned from these two projects. They're in very different places, but there are some similarities. And the first is that climate change is affecting our biggest fisheries. And in some cases, it's interacting with infectious disease. We might see disease as an indicator of stress, as we did with the snow crab, although I could tell you there's another disease in snow crab that we think is actually causing quite a bit of mortality, called bitter crab disease. And we see in the lobster population that this climate disease interaction has caused quite a bit of mortality in southern New England and has raised concerns in Maine. We've also learned that we have more impact when we work with managers. It's been really fun to work with Alaska Fish and Game and NOAA and the Maine Department of Marine Resources. Not only do they help us get access to a lot of organisms, they're out on the water, they see things first. They also work with the fishermen, women to directly. So they help us think about how can we best communicate our work and really have an impact. So it's not just going into an academic ivory tower. We really want to make an impact. We know that multidisciplinary teams are kind of the secret to having these successful collaborations and research projects. We need folks that are modelers, that can model the ocean. We need folks like Melissa that can run the most complex tank experiments. We need students to do a lot of the grunt work sometimes. They all have all their fingers still. So we need pathologists and histologists. So it's really a big team effort, which we rely on federal funding and federally funded programs like the NOAA Trawl Survey to do this kind of work. And then I think this is a big takeaway for us, is that these models allow us to learn from our past and prepare for the future. And I think as we get more and more complex modeling capabilities, we can keep going back in time and learning more and helping to better manage the future. I don't know if we have any more time. Should I wrap it up? 1 minute. Okay, so I will skip over a couple slides, but I do want to say that we are trying to manage these diseases, and aside from sort of the direct management, we're also trying to communicate about this work to a lot of different people of different age groups, because, you know, if we want a behavioral change, we need to have trust. And so we think that is a really big part of this work. And we're doing this at a bunch of different levels. Reyn actually made this beautiful game called Crabdemics. We have a copy up here if you want to take a look, which you should. It's really fun. You get to learn about an epidemic in a population of crabs. You can either play the host or the pathogen. It's super fun, and it's getting sent out to high school students in Kodiak and other rural areas in Alaska, and we actually have 10 copies here, so Maine students get to play as well. We're teaching graduate students. This is a picture from a course that Reyn and I led out in Washington State to students as far away as New Zealand and Japan, and a lot of American students as well, teaching them about methods to study marine diseases. And we're also teaching veterinarians and managers, which is this top photo. Reyn is the star of all the management outreach, but he's talking to a lot of managers and vets in a continuing ed course in British Columbia. And then we're talking to you, trying to tell you about the work we're doing and trying to create some awareness of the importance of this kind of research and the collaborations that we have. So I'll invite Melissa and Reyn up, and we can take some questions.
If I understood, you were hypothesizing that the crash in southern New England of the lobster population in southern New England was a result of the disease. In an earlier presentation a couple of years ago, it was hypothesized that the water temperature was driving a migration as opposed to a mortality event. Comment?
Yeah, absolutely. Thank you. Let me clarify. I think that temperature was the driver of the crash. I think that shell disease happened concurrent with it. And so it's an amalgamation of all these things kind of coming together as one that really caused the demise of the southern New England.
That was great presentation, guys. I'm curious. I know a lot about humans, but not about crustaceans. Do crabs and lobsters have immune responses? And I'm wondering if, like, the increased molting might be an immune response to a pathogen. I just don't know.
Yeah. All right. So that's a great question. Crabs and lobsters do have immune responses, but they don't have the very complex ones that we have. We have immune memory, so we can create antibodies to pathogens we've been exposed to in the past. Crustaceans and other invertebrates do not have that kind of immune memory. What they do have, they can produce melanin, which is kind of the darkening that you see in the shell. That is an immune response. And the molting, yes, it's definitely a defense. We think it's quite costly. It takes a lot of energy to create a new shell. And so one question that's come up for us is what's the balance? Like if you molt to get rid of disease, does it come at the cost of reproduction or growth? So yeah, it's definitely a defense.
Take one more question. Yeah.
Thank you. So the black spot disease, is it transmissible if you put animals that have it together with animals that don't? They transmit it?
This is for black eye. We don't think that it's transmissible, or we don't have any evidence to suggest that it does. So even when we were running our experiments, we had them commingling and there wasn't any kind of seeming kind of crab to crab transmission. So yeah, a big mystery.
First and foremost, I want to thank our staff. This has taken an effort of a small team of folks, including our speakers, and deeply appreciate everything we've done here to make this event happen this summer. I also want to thank HM Payson, who has sponsored this event. HM Payson has been a supporter of the lab and a good partner for the lab over the years. We have public tours on Fridays for the rest of this month. It would be great if you registered in advance, but it's not compulsory. So you can come and somebody will be available to give you a tour. Once again, I want to thank you all for coming. One of the privileges I've had by being here before, having some new experiences and coming back is really appreciating the community that surrounds us at this institution. So we're a very collaborative institution. We have what I would call a very large extended family, and that includes all of you. And so I want to extend my gratitude to all of you and have a nice evening.